Alright, what we're going to do is we're going to finish the Chapter 5, just extracellular messengers discussion today. And then we're going to go ahead and get into Chapter 7. Chapter 7 and 8 give us a real review of the nervous system, which we covered. We crashed course Monday and Tuesday, so it'll seem repetitive, but hopefully it'll begin to start sinking in and you can ask more in-depth questions. But before we get into Chapters 7 and 8, we want to conclude this fifth chapter about extracellular messengers. What are our primary types of extracellular messengers? We're looking at things called, I can hear, but I can't, neurotransmitters, paracrine messengers, what else? Hormones, neurohormones, and what else? What was the other one? The last one was autocrine messaging as well. Of those five, we'll spend our time on the neurotransmitters, the hormones, and the neurohormones. And so this second unit is dealing primarily with our neurohormones. What we learned is, if something's an extracellular messenger, we learned that not all extracellular messengers have the same type of solubility. And we extrapolated from that, well, then they must not all be built the same, nor have the same type of effect as the target cell, is what we concluded basically yesterday. And then we learned that particles that are more hydrophilic must interact at the target cell's membrane with the receptor. And that's where we left off. We said the receptor may just be a simple gated channel. Okay? And that's where we concluded. However, we learned that our hydrophilic molecules may also activate what's called a second messenger system. And that's what we're going to get into a little bit today. Before we get into that, though, our lipophilic ligands, what type of effect would they cause at the target cell? The lipophilic ligands or extracellular messengers do what? Gene activation. Because they activate at the genetic level, the receptors are located where? For lipophilic molecules, the messengers. In the cytoplasm or in the nucleus. But eventually it's going to go into the nucleus. Very good. Let's look in a little better detail about the second messenger systems. They're complex. And because they're complex, they can give us a lot of diversity in response. So a target cell can receive an extracellular messenger, generate a second message pathway, and then it doesn't have to do just one thing. It could do a multitude. And I have a list here, and just like a lot of lists in this class, it's not all inclusive by any means. Okay? It's just giving you an idea what a target cell might do when a second messenger system is activated. Well, one, it might alter plasma membrane permeability, or in other words, change ions across the membrane. But it sounds like what we saw earlier, we're just opening, closing an ion channel. What the difference is going to be is the magnitude. In a second messenger system, we may not just open one ion channel, we may open hundreds or thousands of ion channels. So it's the same type of effect, but on a grander scale. As well as, it looks similar, it might also promote protein synthesis like we saw as a result of lipophilic molecules, except for the pathway to get there is different, and it's going to be bigger, okay, a bigger result. We might turn on particular metabolic pathways, turn them off, may tell a cell to start metabolizing something, tell it to stop metabolizing something. It may cause a cell to release stored vesicles of contents. It just start secreting something that it manufactures. May trigger a cell to divide, okay, or stop dividing, or die, or whatever the case may be. But the catch is, second messenger systems can bring about a wide array of responses at the target cell. And what happens is, it's like described as a cascading effect. Have you used that word, or heard that word before? Cascade, what does that mean? Give me a hand visual. Cascade means what? I got stuck down, okay? Kind of like what's happening. A cascading effect, or maybe you want to use the word like domino effect. Have you ever seen that? Okay, you ever done that when you were a kid, set up dominoes and push them down? Cascading effect. But instead of dominoes or a waterfall or something like that, it's molecules reacting one to another to another. And the point is, the cascading effect of intracellular events are going to cause an amplification in response. So that means is one initial messenger may cause not the production of a single protein, but what? Multiple, or hundreds, or thousands. So instead of 1 to 1, we have 1 to thousands or billions or however many the pathway activates. So it's regulated, but the point is we're going to get a big time response by the target cell by utilizing a second messenger system. We can see this visual of a cascading effect by utilizing this pathway we'll study called the cyclic AMP pathway at the liver in response to epinephrine. Epinephrine is a hydrophilic molecule, and it binds to a receptor called an adrenergic receptor. We'll learn that in a little bit today, but then also in the endocrine system we'll repeat it. So one little epinephrine molecule binds to one little receptor. Instead of causing one little release of glucose, what happens is a cascading series of reactions. And if you look at the quantification, one molecule, I'm going to go 10 to the second power. Activation of intermediate molecules we'll call G proteins in a moment. And then still we begin to increase. Now we go 10 to the fourth power, 10 to the fifth power, 10 to the sixth power, 10 to the eighth power. Increase in magnification. So a single molecule of glucose can actually trigger the release upwards of about a billion molecules. A single molecule of epinephrine can trigger the release of about a billion molecules of glucose. Can you see the amplification? Definitely. And so we're going to look at some of the highlights of how this happens. We're going to talk about some important molecules called G proteins, a second messenger, and these things called kinases before we can get to the response at the target cell. Some important second messenger systems, although not the only second messenger, these aren't it, okay, but these are some of the well-characterized ones. One is called the cyclic AMP. Have you heard of that one? If you heard of one, it's probably this one. You called it CAMP or something like that. There's a very other similar one called cyclic GMP, guanosine monophosphate. Another important one is called the PIP, or the phospholipase C calcium pathway. And another one called the tyrosine kinase. And we can keep going on with the examples of these different types of second messenger systems. We're not going to go through each of them, but we're going to study the cyclic AMP to give an example of how a second messenger system works. And so if you look at your handout, it's going to help walk you through. So we're going to use an image to walk through it, and then we'll fill in the text. So which cell am I at here? What can I call it? Without being specific, I can call it the what? Well, the membrane is the target cell. I know it's a target because I can see a what? Receptor. Okay? And so why don't you go ahead and label this purple thing the receptor. What does the receptor bind? The messenger, the extracellular messenger. And we can actually determine what type of solubility this messenger has. What type of solubility is in this messenger? Hydrophilic. How do you know it's hydrophilic? You both studied it correctly. You said it, though. Someone clarify your its. It. You said this receptor is embedded in the target cell's membrane. You said it, the messenger is where? It's in the extracellular fluid. Okay? That's not diffusing across. So I can tell it's a hydrophilic messenger. Don't use the word hormone. I mean, that's a little bit specific. You call that a hydrophilic It'll bind to its specific receptor. We can already see that the receptor is more complex. It's not just a channel. It's got something associated with it. The thing that's associated with it is called a G protein. The letter G is abbreviated. G protein is what's abbreviated. What do you think the letter G stands for? What do you think the letter G stands for? You look in the image. Not it, so it's a good guess. Not it either. Nothing good sugar on it. Guanine. Where do you get the guanine from? What molecule are we looking at? The guanine diphosphate. GDP. The guanine nucleotide. The G protein binds to guanine containing molecules. In its inactive state that we can see there, it's bound to which? Guanine diphosphate has how many? 2. So it's a little bit of energy, but not as much as maybe a molecule called guanine triphosphate. That's what this G protein does. It binds GDP and GTP. If it's bound to GTP, energetically it has more. If it's bound to GDP, it has less. So when it's bound to GDP, we say it's inactive. What do you think if it's bound to GTP? It would be considered to be activated. What do you think might activate the G protein? What do you think might activate the G protein to become the messenger binding the receptor? Activates it. Causes the G protein to let go of guanine diphosphate and actually bind guanine triphosphate. It takes it from its inactive state to its activated state. Where it's bound to? GTP. Very good. So what's happened is an extracellular messenger bound to its receptor activated a G protein, mobilized it. It's activated, so it's probably going to do something. And the something that it does is then activate an enzyme. Can you begin to see the cascading molecule, receptor, G protein, cascading? The G protein in its activated state activates something called an effector enzyme. So let me back up. Just label this, it says effector, but it's an enzyme. We're just labeling this as an enzyme. The specific name of the effector enzyme in the cyclic AMP pathway is called adenylate cyclase. It's an effector enzyme. So it's an enzyme, it's activated, now it's got to do something. Okay. And so what it does is produces our second messenger called what? And this pathway is called cyclic AMP or CAMP. In order to produce cyclic AMP, ATP is utilized. And produces our cyclic AMP. Notice how on the abbreviation the cyclic is abbreviated with a lowercase c, and the adenosine monophosphate, AMP. That is the second messenger, which means there's more what going to happen? More what? More work. Okay. That's just, what was our first messenger? The hormone or the extracellular messenger. And then it caused the production of a second messenger, which is going to continue on more work, more reactions. It may not feel like it at this point, but really this image is super oversimplified. We're not quantitating it at all at this point. But what cyclic AMP effectively does is activate other enzymes that do what? What do enzymes do? Break down or quicken. Okay? They quicken, they catabolize, or anabolize. They do both reactions, right? Depending on what the specific enzyme is. And so our cyclic AMP tends to activate these things called kinases. K-I-N-A-S-E. It activates these things called kinases. What is the suffix? A-S-E tells us that's an enzyme. And so enzymes do additional what? Work. Just to keep it simple, they do additional work by speeding up specific reactions, whether they be catabolic or anabolic. Phosphate groups. So they transfer phosphate groups. And we have an idea about what happens there, right? When we move phosphates, we change the structure and the function of different proteins. And it activates protein kinase. Is what it does. We still haven't even gotten to the final result inside of a given cell yet. We said this does that and that and that. And the whole point is to tell the target cell to do something. The question is, well, then what does the cell do? We could show that list of 5 things that I said. Well, depending on the cell type, in the example we gave earlier, maybe it's epinephrine at the liver, and it causes glycogenolysis, where glycogen is broken to release glucose. So the liver may cause the release of glucose. Other cells may activate other enzymatic pathways that keep kind of a different process going. Secreting something, opening not just one channel, but hundreds or thousands or more of a cell, or maybe it's a smooth ER, right? Whatever the case may be. There's a lot of variable responses that a target cell might undergo when a second messenger pathway is activated. Some examples of molecules that carry out second messenger pathways are listed in the far left. We'll cover these when we get into really the endocrine system. Some hormones called, you mentioned you knew these yesterday, FSH, follicle stimulating hormone, and luteinizing hormone. Might be familiar with glucagon, parathyroid hormone. They all activate second messengers. And in this cyclic AMP pathway, the big difference is the G protein activates an effector enzyme called what? Adenylate cyclase, which helps produce cyclic AMP, which is our what? Second messenger, whose job is to activate different types of kinases in the target cell, which then do some tangible work within the cell. Let's fill in the handout notes on the right of the image. It'll just review what we just walked through. And I think this is the one where there's an error in the handouts. On the very last bullet, there's a big dashed line. I think that's just an extra dash line. So I think that's where the error is, if I remember correctly. So on that one, you see a big dash line, and panic. Don't panic. Just scratch the little line, okay? So as we identified, we said the ligand, or the first messenger, extracellular messenger, binds to its receptor, which activates a G protein, causing it to unbind guanine diphosphate and bind guanine triphosphate. Step 3, we said the G protein actually then turns on or activates an enzyme called adenylate cyclase. Your book may call it adenylate instead of the suffix ate, and they use YL. The same thing. Adenylate or adenylate cyclase. We okay so far? After adenylate cyclase is activated, it then produces what? It uses ATP to produce cyclic AMP. So it generates cyclic AMP, which we know is the true second messenger. And then in step 5, we identified once the second messenger is available, it activates different protein kinases, which, I have an arrow, brings about different types of cellular effects, depending on the cell type. Liver cells can do something different than a muscle cell, can do something different than a brain cell. Does that make sense? What I did not identify, though, is once that cyclic AMP is produced, to activate the pathway, what should we do? We've got to shut it off. And so the cyclic AMP can be inactivated by an enzyme called phosphodiesterase. So it's an enzyme that gives 2 phosphate groups. It inactivates it. This is a brief introduction into an example of a second messenger system. What questions do you have at this point? Just to put it in perspective, using another molecule, we're familiar with epinephrine, right? It binds to its adrenergic receptors, activates a G protein, which activates adenylate cyclase, which effectively produces cyclic AMP, which activates different kinases. The variation is, what do those kinases do? Well, epinephrine at the liver, we already learned, causes glycogen to be what? Broken down. Glycogen releases what when we break it down? Glucose. It also causes a reaction which we did not study called gluconeogenesis. Gluconeogenesis is genesis, what do you think? Production of new sugars, and glucose specifically. We'll probably come back to that when we get into metabolism. Chapter 6, Unit 3, we'll review that. But effectively, epinephrine causes the release of sugar by 2 different pathways in the liver. If we look at epinephrine on our smooth muscle, we think about where the smooth muscle is, say in your esophagus or your blood vessels, right? Uterus, digestive system. We've got a whole bunch of diversity of smooth muscle in our body. And so therefore, depending on where the organ is and where the smooth muscle is, the smooth muscle may relax under the influence of epinephrine, or in another tissue, the smooth muscle will constrict, okay, or contract. So think about epinephrine when you're running. The smooth muscle in your stomach will probably, do we need to really constrict though? Do I need to digest a lot during running? No, so it causes And my blood vessels, if I want to bring my blood pressure up, they should constrict. So one messenger, one type of receptor, but what would be different to cause the opposite effect? What would be different at the targets? The receptor and which kinases are activated in the messenger. So receptor there are some variations in the receptor, they may all still activate an effector enzyme, but those specific kinases in the target molecules would vary. So causing one to muscle to relax and the other to constrict, even though epinephrine is a messenger. At adipose tissue, epinephrine actually promotes a process called lipolysis, which is lysing lipids, right? So releasing lipids for energy use. And in the heart, if heart rate goes up, it beats faster and stronger. So one molecule, one type of second messenger pathway, but of all the trillions in our cells, in our body, and multiple responses that can happen as a result of the second messenger. And just to drive the nail in the coffin, I've got a little video to help visualize the cascading amplification. I have got the wrong video up. Give me just a second. Okay. So if you look at the second messenger, this video is not completely precise, but it gives us a good visual about how one extracellular messenger binds to a membrane receptor and then activates multiple reactions within it. So we can see the cascading effect. This is some generic looking yellow cell. We can see a huge nucleus, and we can see this little membrane receptor. The effector enzyme in this image is going to be a little blue thing, and the G protein is this little tail region that you see the little tail in there. That'll be like the G protein. What happens is once the first messenger, epinephrine, TSH, whatever, binds to its receptor, it can activate the second messenger system. It produces the second messenger. In the cyclic AMP pathway, the little purple things would be cyclic AMP. Can you see the amplification? It's not just one molecule of cyclic AMP. In this case, we have 6. Each of those can do their own work. So we can begin to see increase in output in this case. And this yellow would be our protein kinases. Okay, so cyclic AMP can activate those, and those enzymes can then go on and do their work. So although it's just showing a few, right, 2 of the protein kinases, okay, add 4 more, and you can begin to see how the response is amplified. So the takeaway is, can you see the cascading? Can you see the amplification? So that's the purpose of our second messenger system. Takes a little bit longer, but the payoff is a big response by the target cell. Continues on. What questions do you have at this point about the purpose of a second messenger system or the example of the cyclic AMP pathway before we move on to good old chapter 7?